Okay, let's dive into this. Welcome, everyone. Today, we're going to explore a truly groundbreaking paper investigating exactly how Huntington's disease destroys one of the cell's most essential recycling systems. Now, Huntington's disease is a devastating neurodegenerative disorder, and for years, scientists have known that it involves the accumulation of toxic proteins that just somehow disrupt cellular function. But the exact mechanics of how this toxicity physically breaks down the cell's internal cleanup crew, well, that has remained a massive puzzle. So in this explainer, we're going to walk step by step through a fascinating paper titled Impairment of Lysosomal Quality Control in Huntington's Disease. We're going to look at the exact evidence, the specific experiments, and the incredible cellular machinery at play. To do this right, we're going to treat this explainer like a scientific detective story. Here's our path. First, cellular recycling in crisis. Second, meet the key cellular players. Third, proving the protein trap. Fourth, testing the clearance machinery. Next, gene expression and membrane damage. And finally, we'll put it all together in the vicious cycle model. Let's get to it. All right, part 1, cellular recycling in crisis. This is the foundation of our lysosomal biology. So let's establish our biological crime scene, right? First, we need to identify the prime suspect, and that's mutant Huntingtin, or mHTT. Huntington's disease is caused by an inherited mutation in the HTT gene. This mutation basically produces an abnormal version of the Huntingtin protein with an elongated polyglutamine or polyQ region. This extra length is a huge problem because it causes the protein to misfold. Instead of floating normally in the cellular fluid, these mutant proteins stick to themselves, forming these massive, toxic protein aggregates inside neurons. But why do these aggregates ultimately kill the cell? Well, to understand that, we have to look at how cells handle their garbage. Think of your own home for a second. Every single day, garbage accumulates. If nobody ever collects it, your house eventually becomes completely unlivable. Your cells face the exact same problem. They constantly produce damaged proteins, worn-out organelles, and protein aggregates. The cellular garbage trucks are called lysosomes. These are tiny, membrane-bound organelles packed with highly destructive digestive enzymes. They maintain this highly acidic internal pH of about 4.5 to 5, which provides the absolute perfect environment to break down toxic waste. But keeping those destructive enzymes contained is absolutely critical, because if a lysosome membrane breaks, those enzymes can leak out and literally destroy the cell from the inside. Because of this intense danger, your cells actually have a brilliant defense mechanism called lysosomal quality control, or LQC for short. And this system relies on 3 crucial R's. First up, repair. If a lysosome sustains a tiny tear, calcium leaks out, and that triggers rapid repair proteins called the S-CART machinery to quickly patch the hole. Second, removal. If the damage is just too big to patch, the cell tags the broken lysosome with a molecular marker called ubiquitin. That triggers its complete destruction through a process called lysophagy. And third, replacement. Once destroyed, the cell obviously has to manufacture a brand new lysosome. This paper asks a really crucial question: does mutant Huntingtin impair the specific 3-step quality control system? Moving on to part 2. Meet the key cellular players. It's time to identify our suspects and detectives. First up is GAL3, or galectin-3. You can literally think of GAL3 as the cell's fire alarm. Normally, GAL3 just floats freely out in the cell's cytoplasm, but inside the lysosome, hidden safely away, are sugar molecules called glycans. When a lysosome membrane ruptures, those inner glycans are suddenly exposed to the outside. GAL3 instantly recognizes this and binds directly to the exposed sugars. When scientists tag GAL3 with a fluorescent marker, those damaged lysosomes light up as bright green dots, or puncta, under a microscope. So this gives researchers a direct visual way to see exactly if and when the lysosome membranes are breaking. Now, let's meet the absolute heroes of our story here, TFEB and TFE3. These are transcription factors. That means they don't actually digest the garbage themselves. Instead, they act as the cellular factory managers. When a cell realizes it needs more lysosomes, TFEB and TFE3 travel straight into the nucleus, bind to the DNA, and turn on the specific genes required to build more recycling workers, machines, and lysosomes. TFEB is widely considered the master regulator here for sure, but TFE3 operates in a very similar, closely related way. So what did the scientific community actually know before this paper came out? Well, researchers already knew TFEB activity was impaired in Huntington's disease, and that artificially boosting TFEB could reduce that toxic mutant Huntingtin. They also knew lysosomes looked really abnormal in sick neurons. But they still had major unanswered questions. Like, was mutant Huntingtin physically trapping TFEB? What about TFE3, which had honestly barely been studied in this context? And was mutant Huntingtin actually tearing holes in those lysosomal membranes? Which brings us to part 3, proving the protein trap. This is where we gather the biochemical evidence. This is where the researchers really put on their forensic gloves. They wanted to know if TFEB and TFE3 were getting physically stuck in those toxic mutant Huntingtin aggregates. Under a microscope, they might look close together, but that doesn't prove they're physically stuck, right? So they used biochemical fractionation. They took cells with normal Huntingtin, labeled 17q, and cells with mutant Huntingtin, labeled 73q, and a severe 145q. They washed the cellular proteins with increasingly harsh chemicals. Mild detergents washed away soluble proteins. Then Sarcosol washed away tougher proteins. But the massive aggregates, they wouldn't dissolve until they used urea, an incredibly harsh chemical. Look at the bottom row of this table. TFEB and TFE3 were found trapped in that highly insoluble urea fraction only when the mutant 73q or severe 145q Huntingtin was present. And this next experiment brilliantly illustrates the point further. They used a filter retardation assay, or FRA. Imagine pouring a chunky smoothie through a coffee filter. The liquid passes right through, but the big chunks get stuck. That's exactly what this assay does. They poured the cell contents through a cellulose acetate membrane with tiny pores. Normal proteins slipped right through, but the massive mutant Huntingtin aggregates, they got trapped right on the surface. And when they checked that trapped material, they found TFEB stuck in both the 73q and 145q conditions. TFE3 was also trapped, particularly in the severe 145q condition. This is hard, physical evidence that our factory managers are getting locked inside the cellular garbage. To be absolutely 100% certain, they brought in a high-tech method called FloIT, flow cytometric analysis of inclusions and trafficking. They attached glowing green fluorescent tags to TFEB and TFE3, broke the cells open, and shot the contents through a laser, one single particle at a time. The laser counted the glowing clumps, and the results were super clear. As the mutant Huntingtin got more severe, the cells formed way more inclusions containing trapped TFEB, and they were noticeably larger. TFE3 also got trapped heavily, especially in that extreme 145q condition. The trap is real, folks. Okay, part 4, testing the clearance machinery. We're moving from just seeing an association to getting actual functional proof. So now we know they're trapped, right? But does it actually matter? This brings us to a really critical question for this explainer. Do TFEB and TFE3 actually help clear mutant Huntingtin from the cell? Because if they do, then getting trapped is a massive disaster for the neuron. To find out, the researchers ran a classic biological test. You add more of something, and then you take it away. First, they forced the cells to produce extra TFEB or TFE3, a process called overexpression. And when they did this, the amounts of both soluble mutant Huntingtin and those big insoluble aggregates significantly dropped. The extra factory managers successfully rallied the cleanup crew. Then they did the exact reverse. They used molecules called siRNA to silence or remove TFEB and TFE3 from the cells. Predictably, removing them made the mutant Huntingtin accumulation so much worse. TFEB's loss was particularly damaging, showing it's a completely critical player in keeping the cell clean. So how exactly are they doing this? Well, the paper infers this happens through a process called aggregates. It works like this. Special cargo receptors, like a protein named P62, recognize the toxic aggregate. The cell then builds a little bubble around it called an autophagosome. That bubble fuses with a lysosome, and the acidic enzymes digest the aggregate. TFEB and TFE3 write the blueprints to build all these different parts. Now, just a quick note on scientific rigor here. The researchers proved that adding TFEB reduces Huntingtin, but they didn't map every single intermediate step of this exact pathway in this specific paper. Still, based on everything we know about biology, this is the highly probable clearance route. That takes us to part 5, gene expression and membrane damage. It's time to read the cellular blueprints. Now, what's really interesting here is that the results were actually quite a surprise. The researchers tested the expression of a whole panel of genes to see if trapping TFEB and TFE3 caused a total collapse of the lysosome factories. But check out the table. Most genes, like the structural lysosome genes, didn't change significantly at all. There was no global collapse. Instead, in the severe 145Q condition, only very specific stress response genes spiked. LGALS3, which makes our fire alarm protein GAL3, shot way up. BAG3, a gene that tries to handle massive proteotoxic stress, also spiked. The cell wasn't entirely shutting down. It was selectively screaming for help and trying to compensate for severe damage. And the researchers could visually see all this damage happening. Under the microscope, they looked at LAMP1, which is a protein that basically outlines lysosomes. In the mutant cells, these LAMP1 structures became massively enlarged and swollen. Next, they looked for that GAL3 fire alarm. Sure enough, in the 6 cells, GAL3 formed those bright green puncta, proving definitively that the mutant Huntingtin environment was tearing physical holes in the lysosomal membranes. But here is the really hopeful part. When they added extra TFEF back into the cells, it significantly reduced those damage puncta. Adding TFE3 also helped, particularly in the most severe cells. Restoring the factory managers literally helped patch the holes. Finally, part 6, the vicious cycle model. Let's put all these pieces together. Based on all this exhaustive evidence we just went through, the authors propose a self-reinforcing vicious cycle. Let's walk through it step by step. First, mutant Huntingtin misfolds and forms those massive insoluble inclusions. Step 2, those inclusions act like a trap, physically sequestering TFEB and TFE3 away from the nucleus. Step 3, without their factory managers, the cell's ability to coordinate repair and replacement suddenly drops. Step 4, damaged, leaking lysosomes start to accumulate, triggering those GAL3 alarms everywhere. Step 5, because the recycling system is now weak, it fails to clear out the mutant Huntingtin, which leads straight into Step 6. More Huntingtin accumulates, forming even more aggregates, trapping more TFEB, and restarting the vicious cycle all over again until the neuron tragically dies. Now, as with any great explainer, we really have to look at this impartially. This study has immense strengths. They used multiple models and threw a powerhouse of assays at the problem. FRA, FloIT, microscopy, qPCR. They successfully highlighted TFE3 as a totally overlooked player, but there are still remaining scientific gaps. They proved the proteins are trapped in the same insoluble clumps, sure, but they didn't prove they physically bind directly to each other. Also, as we noted, the fact that many lysosomal genes were totally unchanged complicates a simple model of total transcriptional failure. Finally, this was largely cellular biochemistry. They still need to prove that boosting TFEB or TFE3 in a living animal actually improves its behavior and survival over time. So the crucial point we're left with today is this. We now understand the trap. We've seen the vicious cycle. But if we're storing TFEB and TFE3 can patch the membranes and clear the toxic garbage in a petri dish, could designing drugs that specifically boost these exact factory managers be the master key to unlocking a future therapy for Huntington's disease patients? It's a highly provocative question, and one that leaves us incredibly hopeful for the next phase of this critical research. Thank you so much for joining me on this explainer, and keep exploring.